Good day, everybody. This is Chris with the Ancient Scholar. I hope this video finds you all doing well. And today what I'd like to do is I'd like to address something that I see making the rounds, particularly on social media, and unfortunately it's being propagated by fellow healthcare providers, and I just don't think people are really looking into this concept in any detail, looking at any of the nuance behind some of the assertions that are being made. And so some of the common assertions that I see are something like SARS-CoV-2 or COVID-19 quote unquote attacks hemoglobin, and people are saying that this is what is potentially accounting for the high rates of morbidity and mortality that we're seeing in some patients who are suffering from COVID-19. So what I want to do is I want to talk about the study where this hypothesis and I want to say that this is in fact a hypothesis, and there is no human data in fact there are no animal data as well. Alright, so this is a hypothesis. There's no human data / animal data to suggest that this is the case. So before I talk about some of the nuance, let's just take a look at the article itself. So here is the article. Let's bring it up. All right, so what do we got? This is COVID-19 attacks, there's that word, the 1-beta chain of hemoglobin and captures the porphyrin to inhibit human heme metabolism. So again, the title of this particular article sounds very interesting and very catchy, of course. These are the authors here. These are two authors that come out of China. One of them is a computer scientist, does computational modeling, and then another one out of the School of Life Sciences and Food Engineering. And that's going to be important because of the kind of study that this is. So here is the abstract to the study, talking about the novel coronavirus, or COVID-19. Let's see if this will let me move it up here. So what I want to do is I want to talk a little bit about the model, or what they did. So what they did was they downloaded the protein sequences from the NCBI database. So essentially they downloaded the protein sequence of the virus itself, some different proteins, and they put these proteins into a model. This is essentially a computer model, and this is actually a whole field of computer modeling known as homology modeling. You can see right there, homology modeling essentially is, let's take the different, and you need to have a fairly precise molecular structure. So you take the molecular structure of the protein, the amino acid structure, and you plot those out, and then you take some other molecules of interest and you go, I wonder if the molecular shape of whatever molecule I'm looking at can somehow interact with these proteins, or these proteins can interact with other proteins. And so you put this into a computer model, and then you run it, and it might give you some intuition as to what to expect. And this can be helpful when it comes to maybe designing drugs or looking at a chemical and go, well, could this chemical potentially interact with this receptor, and gives us some idea of where to focus additional research. All right, so that's really important. That's what they did. This is all computer-based. There is no physiology, no actual observation of physiology in this particular study. And to my knowledge, this study has not been peer-reviewed either, and I'm not saying it's necessarily bad, but it hasn't been peer-reviewed, and so there's still a lot of things that kind of need to happen. So what I want to do is I want to go through. They actually go into pretty substantial detail how they set up their model. which, as far as modeling goes, this is fairly complicated work that was done. But what I want to do, if this will let me, is I just want to move up here and I want to talk a little bit about their conclusions. All right. Again, this is from their paper, from the actual paper itself. Okay. So they're using bioinformatics, but we knew that. So here down in the conclusions, the study, this is really laggy tonight. My internet connection's not great today. So the study results show that the ORF8 and surface glycoproteins could combine with the porphyrin. So porphyrin is a structure within the hemoglobin molecule. It's basically like a bunch of little nitrogens that surround the iron, and then the iron is kind of anchored by an amino acid residue. And so this says that, according to this model, some of the proteins in the virus could combine with the porphyrin, could coordinate and attack the heme on the 1-beta chain of hemoglobin to dissociate the iron to form that forms a porphyrin. So essentially, this would render the hemoglobin unable to do its primary job. And then they go on to say, with these findings in mind, perhaps chloroquine could be of some benefit. And they talk about some of the molecular hypotheses behind how chloroquine could be of a benefit in this situation. And we do know one of the areas where chloroquine works is as far as how it works as an antimalarial drug, is it does work on heme metabolism. All right, so here we go toward the end. So depending on the computational simulation, again, this is a simulation, and discussion of analysis in this study, we speculated the main pathogenic mechanisms of this virus. The virus may first infect, I believe they meant to say infect, not infected, infect cells with ACE2 receptors, including immune cells. Okay, so far this is not outside of the realm of possibility, right? This is fairly standard understanding of how this particular virus works. Immune cells produce antibodies, viral proteins. Antibodies in red blood cells generate immune hemolysis, and this is true, right? In the setting of certain infections, certain severe infections where you have lots of inflammation, hemolysis or breakdown of red blood cells is not uncommon, right? And this is not specific to the SARS-CoV-2 virus. This is seen in lots of different viral and bacterial infections, so this is nothing new. So then they say hemoglobin was infected and then attacked, and I'm not sure. Are they talking about after the hemoglobin gets released? And perhaps that could happen, but at this point, the red blood cell has hemolyzed, it's been destroyed, and so the hemoglobin isn't working anyway, so it really doesn't matter at that point. The virus captured porphyrin and inhibited heme metabolism, but again, the red blood cells hemolyzed, so what does that matter? Therefore, we believe that the damage of the virus to the human body is systemic, not confined to the respiratory system. And there is some evidence to suggest that there are systemic effects, and absolutely are going to be systemic effects because just inflammation is systemic. And then here at the very bottom, this paper is only for academic distribution, right? The correctness needs to be confirmed. Uh-oh, I just lost my paper here. Sorry about that. We've had some really nasty windstorms here in where I live, so the internet is not doing very good today. So let me, I have to go all the way back down again so you can see that this is very, very slow. The good thing is that this is open access. You can look up that link that I've included, and you can look at the study yourself. But again, I do want to spend just another minute or two talking about their conclusion, because they make it pretty clear what they're talking about in this paper, and people that are reporting on this are not. They really did not take the time to look at this and go, oh, okay, this is some computer modeling, this is hypothetical. All right, so in the paper itself, just again, this paper is only for academic discussion. The correctness needs to be confirmed by other laboratories, all right, that can prove whether this theory, really it's a hypothesis, is wrong or correct from the following experiments. Use X-ray to detect hemoglobin in critically ill patients and find out if there are abnormalities, maybe X-ray crystallography or something like that. It is proved by virus experiments, so they actually take the virus itself, not a computer model of viral proteins, but the actual virus itself, and see if it can attack porphyrin and attack heme. And can the viral proteins attack hemoglobin in the blood? Preferably in intact red blood cells, since that would really be where the problem comes into play. All right, so there you go. Right, this is a computer model that somebody did with some assumptions that may or may not be correct. Not particularly high level of evidence, and they just even come out and say, look, this needs to be confirmed, it needs to be looked at, and it's not a conclusion, not a foregone conclusion. It's not really a proper theory. It's just a hypothesis, right? And people are just running with this. Okay, so that's looking at the actual study itself. Let me just add a few closing remarks. All right, so in conclusion, some questions that I want to pose to you guys. So first of all, when we talk about the virus, right, can the virus directly attack red blood cells? Well, remember, what is the job of a virus? Really, from an evolutionary standpoint, a successful virus is a virus that can infect some cell, reprogram or repurpose that cell to produce more viruses, right? It's all about propagating and dividing, right? If you can't do that as a virus, if you can't divide, or you can't reprogram another cell to make more of you, then you've failed as a virus. And viruses that can do that better than you are going to be the viruses that continue and you don't. That's just how evolution works, really. Right, that's just a selection pressure, and they compete, right? So here's the thing. The red blood cell does not have a nucleus. What does that mean? Well, that means that a red blood cell cannot make protein. So if a virus were to infect a red blood cell, say, that red blood cell would be incapable of producing viruses. So from an evolutionary standpoint, why would a virus do that, right? Now, when we talk about other infections, right, like malaria and bacterial infections, that's different, because a bacteria and malaria is a parasite. It's a eukaryotic organism. These are organisms that have their own molecular machinery, their own metabolism inside. They can replicate themselves. They can make their own proteins. And so it would make sense that they could infect red blood cells, because then they could use the red blood cell for food or transportation or something like that. But remember, a virus doesn't have the ability to replicate itself. It needs to infect a cell, and then the machinery within that cell will replicate it. But red blood cells, they don't have a nucleus, they can't make proteins, they don't have ribosomes. This is not a great target for a virus. The next question is, do red blood cells have ACE2 receptors? I'm not aware of any evidence that suggests ACE2 receptors play a substantial role in red blood cells. I could be wrong on that, however. So how could a virus dock to a red blood cell? And then the next question is, well, let's just give these folks, let's just grant you that in fact somehow the virus attacks the hemoglobin and poisons the hemoglobin and prevents the hemoglobin from functioning normally. That produces something known as histotoxic hypoxia, right? So you have a toxicity that results in hypoxia. So we should be able to see this fairly easily, right? We draw blood, we draw an arterial blood gas, we run co-oximetry, right? We look for abnormal hemoglobin. Do we see this, right? Do we see poisoned hemoglobin? Do we see this in COVID-19 patients? We actually don't. What we see in COVID-19 patients is we see patients, particularly these very sick patients, we see patients who have a very low SpO2, right? Or an SaO2, if you're talking about the ABG, right? And they also have a very low PaO2, okay? So what that means is the dissolved oxygen, not oxygen attached to hemoglobin, but the dissolved O2 in the blood, so the actual amount of oxygen that gets dissolved in the blood, right, not attached to hemoglobin, okay, is low, and the oxygen being attached to hemoglobin is low as well. What does this suggest? Well, this doesn't suggest poison hemoglobin, right? Because if it was just the hemoglobin, right, if it was a histotoxic hypoxia, the PaO2 would not be low. In fact, the PaO2 in these patients should be very high because these patients are on a very high FiO2 often case, right? Many of these patients are on 100% oxygen, right? So you should see very high FiO2s, right? You should see FiO2s of 500-600 millimeters of mercury, in the hundreds. We don't see that. We see very low, right? And what does that suggest? A very low PaO2 suggests that there is a problem with oxygen diffusing. So here's the alveolus, and then here's the blood. There's something that is preventing oxygen from diffusing into the bloodstream, right? And that might be VQ mismatch, shunting, those kinds of things, but it does not suggest histotoxic hypoxia. So there are multiple reasons to really think that this hypothesis is spurious, just from a basic physiological standpoint and just looking at these patients. All right, I'm going to go ahead and cut the video off here. Hopefully that you found that helpful. Hopefully this makes sense. And please, if you are a healthcare provider, please look at this video. Please look at the study itself. Right, come to a conclusion that makes sense, and please stop disseminating incorrect information. Right. Just being as truthful and honest as possible is hard, right? Because the situation is changing so much, and what we're learning is changing so much, right? And what we used to say is now different from what we say now. So we need to get this as right as possible, and propagating incorrect information and making claims that are not necessarily made in these actual studies. This just does not help us. It does not help our patients. It does not help the general public. And then another big problem is oftentimes when the first time somebody hears something, whether it's right or wrong, that's the paradigm that they tend to stick with. Even if it is subsequently shown that that paradigm is wrong, and the preponderance of evidence suggests something else, people are often very resistant to changing their minds, right? So they get something stuck in their mind that the hemoglobin's poisoned. Some people will be highly resistant to changing their mind, and that's very problematic, right? Misinformation is a big problem, and the onus is on us as healthcare providers to be as truthful and as honest as possible. And in that vein, I'll actually be doing some update videos talking about how I have changed my opinion given new data on what we're seeing with patients, with COVID-19 patients. And I have alluded to that in prior videos, particularly when it comes to the respiratory management. And I have another video in the queue talking about this because it is so important. Okay, guys, hopefully you found this helpful, and we'll